Yep, I'm Leslie Weston. I'm a professor of plant biology and natural products chemistry at Charles Sturt, and I've been working in photosensitization for about 20 years on and off, and natural products chemistry for about 30. I and my lab, which is a large team here at CSU, focus mainly on plant interactions with plants, pests, and grazing herbivores. And so tonight we're going to talk about photosensitization, particularly cases that have broken out in Northwest Victoria. And I want to present to you a bit of information about these cases, their impact on thoroughbred horses, and also talk about the photosensitization process itself, because many of you, while you may have seen photosensitization, probably haven't seen primary photosensitization, such as in this case. So just to classify different types of photosensitization, there are four main types, and generally here in Australia, we see secondary photosensitization, and in some cases, but rarely, primary photosensitization. You all would be familiar with the concept of photosensitization if you ever took tetracycline as an antibiotic, because this particular type of acquired photosensitization can be brought on by drug use, and it's also light-induced. So primary photosensitization, the one we're going to focus on tonight, is a light-induced dermatitis or skin condition caused by the generation of photodynamic molecules or chemicals. And these chemicals can induce this photosensitization either by direct contact from an organism such as a plant, a fungi, a bacteria, or other organism, or by translocation through ingestion of plant material or fungal hyphae or other organisms or portions, bacteria and fungi, that would be translocated and producing these chemicals throughout the horse, primarily through the bloodstream. Now, primary photosensitization has seen regularly in the US, England, Europe, and occasionally here in Australia and New Zealand. Some of the plants that cause this are giant hogweed, buckwheat, and a number of legumes, including burr clover, other medics, lucerne in particular, which we're going to talk about tonight. The secondary types of photosensitization also result in acute dermatitis, sometimes looking quite similar, but this is created by a different concept. This is caused by phytoporphyrin accumulation due to microbial activity in the gut. And what happens here is that chronic or acute liver damage due to the result of other secondary products in plants, such as pyrrolizidine alkaloids, actually result in liver damage. And then the accumulation of these phytoporphyrins cause this secondary impact or photosensitization. Now, primary photosensitivity in animals really can lead to some really severe dermatitis, secondary infection following on from this primary dermatitis, and even death. But here in Australia, it's rarely encountered. I've been studying primary photosensitization now for about 10 years, and we had a number of cases crop up due to the grazing of bicerula, an introduced legume from the Mediterranean region in Europe, and this causes primary photosensitization in sheep grazing them. What happens here is we have a heightened reactivity of skin cells and dermal tissues upon exposure to sunlight. And what's required for this to happen is the actual ingestion or direct contact of the organism, the grazing animal, with the natural products that are light reactive. So this requires the presence of a light absorbing or photodynamic molecule, and I'll talk to you about what those are in a minute. The reaction is most severe, if any of you have seen this, in white or light colored non-pigmented skin or coats. So for example, merino sheep rather than dark-coated sheep, and in the case of horses, those that have white markings in particular. Following exposure to light, which includes both UV and visible irradiation, we get very rapid responses. So UVC is typically not implicated, but UVA, B, and visible light are. So what happens here is an illustration of some of the types of molecules that cause this primary photosensitization. And you can see from looking at these structures, while they're complicated, they have some things in common. And these include that they're polycyclic, they have oxygenation, and they're large complex molecules with conjugated bonds, which allow for photodynamic reactions to occur. The one on the left, rhodexin A, is actually a fungal metabolite. Phosphorbide A is found in high concentrations in lucerne and other legumes, and generally all plants, but some of them contain and produce more under certain conditions. These molecules can cause damage to the skin, dermatitis, keratosis, and swelling, and more importantly, over the long term, they can also damage DNA, which makes it really difficult for the animals to regenerate new tissues. Insects are also known to produce occasionally some of these primary photosensitizing agents. So here's an example of giant hogweed. This is a weed that I worked with in the United States while I was at Cornell University. It grows along highways. And what happened here is these plants produce a variety of metabolites known as furanocoumarins. They're complex polycyclic molecules that are oxygenated. When you brush against this plant, as do many Department of Transportation or highway workers, because this weed was growing along roadsides all through New York State and the Northeast, contact dermatitis occurs, and what you see is a distinct damage, dermatitis, burning, tissue degradation, necrosis, secondary infection, and a number of our workers actually lost their legs or arms. So it can be extremely penetrating and damaging. In the case of legumes and livestock that graze them, primary photosensitization can be occurring fairly readily from the consumption of lucerne, bicerula, burr medic or clover, and other medics. And primarily, these are the ones that we see implicated here in Australia. So for example, what happens in this case is we have a sheep or a horse, which will graze on the legume itself. They'll absorb a large concentration of secondary plant products, perhaps phosphobides or other chlorophyll related molecules, the porphyrins. These molecules will be absorbed into the bloodstream and transported throughout the animal. Generally, the molecules tend to coalesce or congregate in the extremities. So this would include the nose, the ears, the eyes, the feet, the legs, and the tail area. When sunlight hits the animal, the molecules are photodynamic, so they become reactive. They form free radicals, which could be singlet or triplet oxygen. These free radicals destroy membranes within the dermal tissue, and we have the photosensitization, which can occur within 24 hours of consumption or exposure to these plant parts. And so here's an example of a sheep with photosensitization. When we look at what happens in the animal, and this includes a horse, a sheep, or other cloven hoofed animals, particularly cattle, we see that these complex polycyclic molecules can be translocated throughout the bloodstream. They can also cause contact activity from direct exposure. Here we see ears, eyes, in particular, that are affected in sheep. And in the case of these horses, basically light-colored pasterns and legs. In Northwest Victoria in the autumn of 2020, there were a noticeable outbreak that occurred. This outbreak was reported to myself eventually, along with veterinary specialists at Vic DPI, and 2 thoroughbred studs were involved and 1 paint horse property. all in Northwest Victoria. The properties were about 100 kilometers apart. Photosensitivity was noted over a 6-week period. And during this time, the weather was quite interesting. There was above average rainfall. If typical rainfall during this time of year might be 25 to 30 mils a month, there was 104 millimeters in April after a prolonged drought. So there was moist soil, humid conditions, dew produced, and sunny days. Horses grazed predominantly lucerne pasture, which was about 75% or more established stands. The lucerne was lush, the mornings were dewy, sunny afternoons occurred. The horses were also supplemented on pellets and sometimes hay. There was severe dermatitis noticed with lesions, ulceration on legs, feet, and face, depending on the property. About 32 animals or upwards of 50 to 100 impacted, or in a smaller case, just 4. Blood work and biopsies were performed on at least 5 mares by Vic DPI and local veterinary service providers, and examination of the 4 specific pastures on one farm revealed a very heavy infestation of black aphids, which were later identified as Cowpea aphids. So here's some of the visualized issues in terms of the contact or translocated dermatitis that was caused by this outbreak or photosensitization. And this occurred in one farm in Northwest Victoria, along with one that's about 80 kilometers away, and then a paint horse stud that had a few. And in this case, we see that mares or yearlings, youngsters, that had white markings were typically most heavily affected. So 72% of the 32 mares that were out grazing on lucerne were affected. And this included the dermatitis seen on the pasterns and legs, on the face, ears, and around the eyes, and particularly the muzzle. Mares with no white markings typically were less affected. 5 out of 32 were not colored, and they were unaffected. All stock in the paint horse stud had obviously white coloration, so all were affected. And at a second thoroughbred farm, approximately 50 out of 100 were estimated to be affected. So the types of things that you would see here were dermal changes to blood vessels, dermal swelling and separation of dermal layers over time, hyperkeratosis or this crusting appearance, secondary infections. The horses had applied bandages and antibiotics, but still that didn't prevent secondary infection. And after treatment, eventual improvements were noted following allowing air drying. What you might say would be similar to receiving a 1st or 2nd degree burn with some scarring and eventual disfiguration. So if we look at one mare here over a 6-week period, these photographs were provided courtesy of the stud. We can see contact dermatitis, which was noted in early April. And then within days, this particular individual had a progressive effect in that the dermatitis increased and additional layers of skin were affected. And eventually, after 6 weeks, keratosis and this crusting occurred. Now, at the paint horse stud in Northwest Victoria, this was the type of response that was seen, again, to non-colored areas of the legs and feet, around the eyes, and on the face, particularly the muzzle. So on-farm actions following detection at the thoroughbred studs occurred fairly rapidly, but it was difficult to diagnose initially exactly what was occurring. And so mares were immediately removed from paddocks. They were placed in stables and the affected areas were bandaged. Secondary infection was noted, so bandages were eventually removed and later air drying was allowed. Biopsies of certain horses were performed by veterinary staff. Eventually, primary photosensitization was diagnosed. When sheep were put in the same paddocks after horses were removed, they also experienced some primary photosensitization. When paddocks were carefully examined, it was found that, as you can see from these pictures, the affected plants, this is lucerne, some of them had upwards of hundreds, if not thousands, of Cowpea aphids on them. So paddocks were rated for lucerne establishment. Each of the 4 had greater than 75% lucerne, and all 4 were heavily infested with Cowpea aphids. Pastures were eventually treated with insecticides for aphid control. So what are Cowpea aphids? They're shiny black aphids. They're quite large. You can see them easily. They've got black and white legs. My husband's an entomologist, so he helped me actually prepare and study the literature. And we found that Cowpea aphids in Australia are prevalent in meadows, vetch, and a wide range of legumes, including lucerne and winter pulse crops. They're most active typically in the spring and autumn, but they can be active in Australia all year round. The adult winged aphids are the movable ones, and they often move large distances by wind from alternate hosts to other areas. The hosts are typically legume or non-legume crops and pastures. Occasionally, they can be on perennial plants as well. They reproduce in really large numbers when seasonal conditions are appropriate, and they tend to reproduce asexually, but they can reproduce both sexually by laying eggs as well as asexually. And occasionally they can overwinter as active adults or even eggs. So they can be very prevalent, and their outbreaks in Australia have been reported as early as the turn of the century. So one of the interesting articles I found recently is a correspondence in 1955 to the Australian Veterinary Journal. And this was reported by a scientist who was a veterinarian and working in northern West New South Wales, who found that he experienced and noted photosensitization in guinea pigs, but also in grazing livestock. He worked with Medicata denticulata, which is burr clover or burr medic, and he noted that this was a very strong response. He also noted the presence of aphids within this particular region. He believed that this response was caused by ingestion of aphids, but it was difficult at this time for this individual to try to determine the chemistry of what was going on. And in this case, as he states, one of the most critical tests of this hypothesis that aphid pigments will photosensitize would be the demonstration that aphid pigments occur in the serum of photosensitized animals. To our knowledge, this has not yet been reported in Australia or really in pretty much any literature around the world. So there's quite a bit of research that could be done. So the compounds that are found in aphid species, and noting the literature, there isn't that much reported, but the first real paper was occurring in Nature in 1948 by a group in Cambridge University. They noted a large polycyclic molecule with a lot of oxygenation, named these compounds erythroaphins. There are multiple compounds that are produced within aphids. They studied them in 12 species of aphids and found them in all. In 1955, we found that in the Australian outbreak, the paper I just reported before, photosensitization in burr clover infested by Cowpea aphids. And in more recent literature in the 1960s and 70s, several related metabolites were reported to be present. So interestingly enough, we do find that the aphid may produce some of these photodynamic molecules itself. So Cowpea aphids are interesting. They cohabit the plant with other insect predators and fungi. And what happens on the plant itself is, with thousands of aphids on it, honeydew, which is ingested from the plant by the aphid, is released to the surface of the plant. And often we see very high levels of fungal infestation on these legumes as well. So photosensitization was noted in 2020 in South Australia, Victoria, and New South Wales. And it affected not only horses, but cattle and sheep and goats. And in this case, Cowpea aphid was present, but other aphids were also available during that year and also caused some outbreaks of photosensitization that were reported. Generally, the photosensitization always occurred in association with the legume. And as I mentioned, the aphids, which are produced and live off these host plants, produce this honeydew, but they may also produce their own complex of photocytotoxins. So the fungi survive on the plants with the aphids and the honeydew, and also can frequently cause photosensitization, and that's reported widely in the literature. So here, in this case, these horses might have been exposed to photosensitizers in the plant, in the aphid, and also in the fungi. So it could be the perfect storm. So how best to manage the situation? As farms noted, really important to remove the affected animals immediately from intense UV visible light exposure and from the infested pastures. Contact activity could be the cause, but translocation from eating these ingested plants could also cause it. Biopsy should be performed if in doubt. Place the animals in covered stables, treat the wounds as if they were first or second degree burns, but apply antibiotics and bandages if necessary, and then eventually, as the animal progresses, air dry to allow for drying and potential improvement. At the same time, treat pastures for cowpea aphids as needed with broad spectrum insecticides. Monitor the pastures regularly. Annelies is going to talk about this. Very important to look at wind, adequate moisture, and healthy plants. If all of these things are there, it can result in devastatingly high aphid numbers. Why a cowpea aphid? Well, in this case, it's a really large aphid. It eats very voraciously. The questions that arise are, does the aphid harbor photodynamic molecules itself, or does it actually transform the molecules that are plant or fungal in origin? We don't really know at this point. So we have opportunities at Charles Sturt University now to identify the causal chemical agent and metabolite. And we would like to do that by seeking funding for this. It's an interesting challenge, but it's an important one because lucerne, as well as a whole number of other legumes that horses ingest, along with grazing livestock, are evidently integral to this interesting challenge and devastating possibility for the racing industry, particularly when raising yearlings or mares that are in foal. One of the questions we need to ask is the plant, aphid, or fungus the source of photodynamic molecules? We actually can't say that for sure. It may be all three, it may be one that predominates. Are the metabolites transformed by the cowpea aphid? We don't know that yet. How is the causal agent transferred to the grazing horse? Is it direct contact or transported through the bloodstream, or both? We would suspect both. The causal agents are molecules that are polycyclic. We're certain of that. And they're likely complex ring structures that undergo serious oxidation or transformation when exposed to light, generating singlet or triplet oxygen. Their transformation in the presence of mixtures of compounds could result in increased potency, such that the 2020 outbreak was unusual because of the high level of photodynamic molecules and their potential to transform. So clearly, more advanced analytical techniques are available today than 50 years ago, and we should be able to relatively quickly address this issue clearly. But I note it's quite important to review the older literature. So what we would potentially do is extract and perform metabolomics. We're capable of doing that and frequently do use this to identify photodynamic molecules, such as the ones located in Bicerula and causing outbreaks in sheep. We have assays to assess the photodynamic molecules activity in epidermal cells, and we can see that in most cases with grazing legumes, the highest concentration of these molecules are in leaf tissue, with next most common stem, and then less in roots or other portions of the plant, including inflorescence. The lightning color is an indication of cell death. And so when we have higher concentrations, we get increased cell death. And many of these photodynamic molecules are able to be extracted relatively easily from plant tissues using ethanol. So the tool of trade for us here at CSU are a lot of analytical equipment. We have a warehouse of them. These are Agilent mass spectrometers. We perform metabolomics to relatively quickly identify, isolate, and quantify these compounds. And we work with a variety of chemists, veterinarians, and animal and human physiologists to do so. So just to get back to the horses that were impacted in this case, what's actually happened to them? Well, in following up, this outbreak occurred over approximately 15 to 18 months ago, and an update on the affected individuals. Those with limited injury, like the mayor up at the top of the page, recovered, but those with deep dermal lesions and many layers of dermal tissue impacted have seriously impaired damage in terms of permanent scarring and potential damage. All of them luckily are still sound, but they will consequently bear the results of some of this for a lifetime. So one of the things we have to really look at is how this might impact the sale of yearling stock or weanlings. The other thing that we'd like to look at is if legumes that are dried or conserved, such as silage or hay, also have the opportunities to cause this particular type of photosensitization. There have been reports in the literature that they do, but to a lesser extent. I'm pleased to present my research associates in natural product chemistry, Dr. Russ Barrow, Dr. Sal Gurusinghe in microbiology and cell physiology, Dr. Paul Weston, an entomologist who also does metabolomics and mass spectrometry, which is very handy for this project, Dr. Sajid Latif, who works in veterinary physiology and bioassay development, Dr. Haley Randall and Sharan Radal in the School of Ag, Environment, and Veterinary Science, working in the equine physiology and behavioral aspects. And all these future studies are proposed to be conducted at Charles Sturt here in our National Life Sciences Hub, and with cooperating veterinary clinical center here and cooperating farms and studs. So with that, I'd like to acknowledge my research supporters who work with us in photodynamic molecules before, the Meat and Livestock Australia, the Graham Center for Agricultural Innovation and Charles Sturt University. And I'd like to acknowledge Ag Victoria for their assistance in providing biopsy results and also assay results. And looking forward to hopefully working more with AgriFutures. And also Dr. Claire Williams-Patterson, who was integral in studying some of these outbreaks.
Thank you, Leslie. Very insightful. I definitely, I remember when that came up, obviously last year there, but we do have a question.
Yep.
Mark, we've got two here. Mark said, great presentation, thank you. Is there any, oh, I just lost it now, 2 seconds. So is there any research on the efficacy of mycotoxin binders added to the feed of horses who are suffering, I just lost that again as we were showing there, who are suffering from photosensitization?
Not much that I've seen reported. We've worked with mycotoxin binders in a couple of cases where we were looking at, for example, endophyte toxicity, and worked on development of therapeutic tools for example, staggers, ryegrass staggers, but much less done in the case of primary or secondary photosensitization. It's an interesting idea. In this case, probably wouldn't be as impactful because of the levels of molecules that would have had to be ingested to cause this type of outbreak, but interesting nonetheless.
Perfect. And we have one from James. Other than the photosensitization, were there any other systematic abnormalities reported in affected horses?
In the biopsied horses which had blood work done, no, there actually weren't. And so they were normal in all other aspects. But Emma, you might want to come on and have a go at this question too.
Yeah, hi James, thank you for your question. Look, we didn't really see any other changes. We had 65% of these horses were in foal, and fortunately the pregnancies were not impacted at all. But obviously it was quite a concern having two stable mares, and there was a lot of inflammatory responses as well, obviously, but no other systemic abnormalities that we could identify.
Okay.
Excellent. Thanks. Thanks, Leslie. And thanks, Emma. So for those of you who don't know, Emma works at one of the impacted studs in northeastern Victoria. So we asked her specifically to come on just in case you had any specific questions for the stud. So thanks for coming on.
No worries. Thanks for having us, Annelies.
That's okay. So I just really briefly wanted to talk about, because for those of you who don't know, which would be everybody, my background is actually in pasture agronomy. So I was able to provide some value here around actually how to go about checking your pastures for Cowpea aphids. So I also spoke to a good friend of mine in southeastern New South Wales on the Monero, where they'd had some issues at the same time around Cowpea aphid photosensitization in sheep. And so we got together and sort of put this slide together. So as part of this, we put together a document, which I will add in the chat after I've finished, which gives you a really good background on how to do this, how to check your pastures and what to do if you find it, etc. And then if you have any questions about how to treat your pastures for Cowpea aphids, I would highly suggest that you talk to your local agronomist at your local rural supply store about what would be the best, because there are grazing withholding periods associated with these insecticides that you'll need to take into consideration. So thanks to Leslie, who had some really great pictures of the Cowpea aphid, probably better than mine, but they are quite a small, large for an aphid, sort of about so big, little grey little insect with these black and white legs, which makes them quite distinctive. And they have a sort of grey colour, a dull grey colour, which is actually because of the sort of wax on the outside of the individual. It makes them the grey, but when they're adults, they turn really quite shiny black. And then as they mature, they grow wings and can be quite mobile across your paddocks. So the times to really look for these insects are coming up very shortly. So definitely through that September, October, November, and then April, May, and June. So very soon, I would suggest that if you have a stand that's a heavy lucerne stand, I would suggest that anything over 50% lucerne, that you would start really heavily monitoring these paddocks for Cowpea aphids, especially in Victoria. But if there was a high load last year and there's been significant rain this year over winter, as there has been, I would suggest that this could be an issue across most of the thoroughbred areas within Australia. So the best way to do a paddock inspection is to start at a different point each time and to inspect a paddock at least once a week. And what you do is you just pick a point and you walk towards the center of the paddock and you pick out 20 plants at each point. Now, they don't need to be the full plants. You don't need to pick them out of the soil. Get a good stem and have a look. And you should be able to, with bare eyes, you won't need a magnifying glass or anything like that, see if you have any aphids. There will be other aphids running around. Often there'll be red-legged earth mites also going around at this time of the year in lucerne and clover pastures, and also on flatweed or dandelion as well. But don't be concerned about those. But if you do start to identify some Cowpea aphids on your pastures, I would suggest removal of the horses immediately. As Leslie noted, because we're not sure if it's if it's the aphid itself, if it's how the impact it has on the pasture, or if it's anything to do with a fungi associated with it. We do suggest removal from the paddock, and even possibly removal until well after the paddock has been sprayed. So it's a bit complicated. Your local agronomist will absolutely help you out to help you understand how to do a paddock inspection. But I will also pop in the chat this document that we put together, and hopefully that gives you enough information to be able to find them and then what to do. And if you've got any questions, Leslie, myself, or Emma would be happy to answer them. Thanks.